Now, going back a little bit to the history of microbiology, a scientist named Kohn was the first to classify bacteria based on shape. And that's a system that we actually still use to this day. So there are circle-shaped bacteria, rod-shaped bacteria, spiral-shaped bacteria, and we still use that classification in microbiology. He also recognized that a genus called Bacillus could produce spores, and these spores allow the microorganism to survive a lot of changes to the environment. So it allows them to survive being dried out, lacking nutrition, too hot, too cold, antibiotics. And one of the most famous bacteria that does this is Bacillus anthracis, which causes anthrax. And then Joseph Lister, he found that microorganisms also led to spoiled milk. So the reason milk goes bad is because of microbes. He did a lot of other things as well. We'll talk about him a little bit more, but he's a pretty famous microbiologist. As we start to get more, towards the present, there were scientists who finally discovered evidence of a virus. They found that an infection could pass through a filter, and the filter should catch things like cells. It's a pretty small filter, but still the infectious agent was in there, and that was our first evidence of a virus. And then scientists figured out that insects could transmit bacteria too, or arthropods. So the first bacterium identified to be transmitted by ticks causes a disease called Texas cattle fever. So once, cell theory started, so 1830s or so, the rapid expansion of our knowledge of microbiology was really significant. And I think it's like that with lots of different discoveries. I mean, many of you are probably a lot younger than I am, but when I was a kid, we didn't have dial phones. I'm not that old, but our phone was like corded. We didn't have a cordless phone, so you had to sit there with the cord and talk on the phone. And then we got a cordless phone. They were more expensive, so we waited until they got cheaper. We got a cordless phone. And then I had this big brick of a cell phone that was actually my parents' because I didn't have my own. And now I have a phone in my pocket that does more than, my first computer could ever do. It's got way more storage. It's faster. It takes pictures. It's amazing. So we see the same exponential increase in knowledge in science as we do in technology. And I think that's really cool. In 1928, a scientist named Griffith discovered that bacteria have the ability to transfer some characteristic about themselves to other bacteria. And that becomes really important when we talk about antibiotics and antibiotic resistance. So bacteria are able to exchange what we now know is DNA, but at the time they didn't know it was DNA. And this, if you ever take a science class, like a genetics class or a microbiology class, this is a really, really classic experiment in microbiology and genetics. In the 1940s, scientists began to learn that genes would code for enzymes, but they still didn't know if genes were proteins or if genes were DNA. But they knew that bacteria could pass genetic material, regardless of what it was. And they identified that bacteria could mate. So bacteria can exchange genetic material. So it's not mating in the classic sense of sexual reproduction, but bacteria can make this structure. It's called a pilus, P-I-L-U-S, and actually pass genetic material from this cell to this cell, which again has really important implications when we start talking about antibiotic resistance. In the 1950s, scientists identified that viruses could also move DNA between bacteria. So bacteria can exchange with themselves, but viruses can also help move DNA between bacteria. And in 1953, that's when the major papers were published confirming that DNA is the genetic material. So DNA is what gives us proteins, DNA is passed from parent to offspring, etc. So these were Watson and Crick. They published the major paper describing the structure of DNA and proposing how it might replicate and things like that. This is Rosalind Franklin. She was a X-ray crystallographer. She took X-ray pictures of very small things. And so this is a picture from her lab showing, I'm not an X-ray crystallographer. Don't have the ability to actually interpret these very well, but this is what allowed them to figure out that it was a double helix, that we have that nice double helix of DNA. And so Watson and Crick won the Nobel Prize for their discovery. Franklin probably should have won. Okay, let's rephrase. Franklin definitely should have won as well. They can give it to 3 people at a time. But she had died prior to the Nobel Prize being awarded. And you can't win the Nobel Prize if you're dead, or you can't be nominated if you're dead. I think one person, maybe 1 or 2 people were nominated and then died right before it was actually officially awarded. But you can't be nominated if you've died. And so she had died of cancer caused by X-ray exposure. So her boss won the Nobel Prize with Watson and Crick. In the 1970s, we started making even more discoveries about microorganisms. Scientists identified viruses that could turn RNA back into DNA. We call them retroviruses. And in the 1980s, we found one of the most famous or infamous retroviruses. But this phenomenon was really unique because for the longest time, scientists thought that DNA could be turned into RNA. Not turned into, but there could be an enzyme that would make RNA from a DNA template, and then they would make proteins from the RNA template. But now we found an enzyme that can turn RNA back into DNA. And also that bacteria have these pieces of DNA that are outside of the chromosome. We call them plasmids. This is what bacteria usually share with one another. And in the 1970s, scientists discovered archaea, which are these extremophile organisms that live in deep sea thermal vents and hot springs. So for a long time, people thought those environments didn't have life, but it turns out they actually do. In the 1980s, scientists discovered prions. Those cause mad cow disease. So they're misfolded proteins that can cause the brain to deteriorate. So that's what you're seeing here. This is a normal brain, and this is a prion-infected brain. Scientists characterized HIV. So in the 1980s, HIV was really decimating populations. And so it was really important to figure out what it was so we could try to figure out how to stop it. And so they characterized the virus as a retrovirus that turns RNA back into DNA. And scientists developed a technique called PCR to allow us to make lots and lots of copies of DNA. And this technique is so important for pretty much everything we do in genetics now. It also won the Nobel Prize, but this is why, you can identify a criminal from a drop of blood, or you can easily do paternity tests, or you can see if you have a mutated version of a gene, or any number of things. So this is super, super common now. In the 1990s and 2000s, scientists really began doing a lot of genome sequencing. So the first organism that was sequenced was a bacterium called Haemophilus influenza. It was like I said, a bacterium, pretty small genome. The next year, they published the first eukaryote, so the first thing with a cell. It's brewer's yeast or baker's yeast. I'm sorry, the first thing with a nucleus. That's a eukaryote, remember? Nucleus. The first animal, even though they don't look like animals, they're animals, was a nematode. The first insect, a fruit fly. The first plant is called a Rapidopsis. It's a very common model organism plant. And the first human draft was published in 2001. So we have learned a lot about life on our planet within the past 20, 30 years because of our ability to sequence these organisms. So what is the future? Well, the future is another Nobel Prize winning discovery, CRISPR. Being able to edit people's DNA. So now we know that the DNA is there. We know what the DNA does. We know what happens when the DNA is changed and it shouldn't be. So when you have a mutation. And now we know how to fix it. Do we know how to fix it in people on a really big scale right now? No. But is that the future? Maybe. And why is this relevant to microbiology? Well, because CRISPR was actually discovered in bacteria. And it's the way that bacteria fight a viral infection. So scientists learned how to manipulate this natural phenomenon in bacteria and use it to edit the DNA of any other organism.